Welcome, everyone. Wow, what a topic we have today. We are going over genetic variation. I mean, this is cool stuff, y'all. Meiosis and mutations will be our main focus, so get excited about going down to the cellular level and discussing the reasoning behind the differences in traits among us. So, my brown hair and blue eyes and hitchhiker's thumb are totally ready for this. Science rocks.
All right, open up that biology Google Drive or your notebook and get ready for some good notes and sketches today. You might grab some colored pencils too while you're at it. So copy down these 39 vocab words. They go in ABC order like this, and just come back later and create your own definitions of these words. Remember to always use your own words and draw pictures. Make sure to draw the steps of meiosis and label that out for sure. So you can pause this if you need to finish writing down the terms. All right, during the process of meiosis, the number of chromosomes in a cell is actually reduced by half. So, meiosis is chiefly involved in the production of sex cells called gametes. All right, so makes sense, right? If you get 23 chromosomes from your dad and 23 chromosomes from your mom, then it adds up to be 46. All right, so here are the phases of meiosis from interphase all the way to cytokinesis. I remember this prophase, metaphase, anaphase, telophase part, and then there's stage 1 of that and then stage 2 of that. I remember the order of those by PMAT. And PMAT on its own, I mean, it means really nothing, although I think of a puppy and a PMAT. So PMAT helps me remember the order of prophase, metaphase, anaphase, telophase. So maybe PMAT can help you as well. So let's look at these phases. It's going to start up here at interphase, go all the way down through cytokinesis. Okay, so we'll start right here. I'll just put an I for interphase. Chromosomes are copied during interphase, okay? This short period of interphase is also known as the S phase for synthesis. It's making or copying these chromosomes, all right? So during prophase 1, I'll just put a P1 here. All right, during prophase 1, homologous chromosomes pair and become something called tetrads. There are 2 chromosomes, or really 4 chromatids. So if you look at the picture, you can see where they're put together. In interphase, it's all kind of a squiggly mess, and in prophase, the homologous chromosomes pair. Crossing over between homologous chromosomes occurs at this stage, okay, in prophase 1, crossing over. After crossing over occurs, homologous chromosomes line up along the equator, or like the middle, in metaphase 1. So I'm going to put an M1 for metaphase 1. Okay, think meta, middle, metaphase 1. Whole chromosomes separate from the tetrad formation and move to opposite sides of the cell. See how they're separating, moving to opposite sides of the cells. They started here and they're separating apart. That is anaphase 1. Okay, each chromosome still has two sister chromatids, okay? So you can see right here, each chromosome has 2 sister chromatids, but they're separating, okay? During telophase 1, down here is telophase 1, during telophase 1, a nuclear membrane forms around each set of chromosomes. So you can see this dotted line here. So they pulled apart here, and then this nuclear membrane forms around each set in telophase 1. Each cell now has one set of chromosomes. Okay, so each cell has one set of chromosomes and is haploid. Okay, haploid, also known as N. Okay, haploid. So each cell has one set of chromosomes. Sister chromatids become short and thick at the beginning of prophase 2. So we're back up here, and this is prophase 2. Okay, the chromosomes migrate to the center of the nucleus and line up along the equator or the middle in metaphase 2. Metaphase 2. Okay, so remember meta, middle. Okay, so remember we have two separate cells now, meta, middle. They both line up. During anaphase 2, so we're right here at anaphase 2. During anaphase 2, the sister chromatids are pulled apart. Okay, the sister chromatids are pulled apart by microtubules to the opposite poles. Okay, so they're pulling apart to the opposite poles. All right, and then we have a nuclear envelope forming around each set of chromosomes in telophase 2. So down here we have telophase 2, where that nuclear membrane is forming around each set. Okay. After these meiosis steps, cytokinesis. Yes, where it finally pinches into its own little cell. Cytokinesis takes place. During cytokinesis, the cytoplasms and organelles are divided among the 4 haploid daughter cells that contain only half of the species' usual number of chromosomes. Okay, so each one of these, remember, has 23 chromosomes. You get 23 from your dad, 23 from your mom, and then fertilization will make 46. Okay, so meiosis and fertilization. At the top of this diagram, a special cell that undergoes meiosis to become a male's sperm, that's the gamete, is shown on the left. So this special cell on the left undergoes the steps, and each one produces a male sperm. And another cell that undergoes meiosis to produce a female egg is shown on the right. So here's a special cell, undergoes meiosis, and each one forms a female egg. Okay, so sperm cells and eggs are known as haploid cells because they contain half the set of genetic information. Remember, each one of these has 23 chromosomes in a sperm, and each one of these has 23 chromosomes in each egg. So, however, in the final step of this diagram, all right here, in this final step of the diagram, when a haploid sperm combines with a haploid egg cell, fertilization occurs. Fertilization occurs, and a diploid offspring is formed. Okay, so now this diploid offspring contains a full set of genetic information. 23 chromosomes from the sperm, 23 chromosomes from the egg, adds up to be 46 chromosomes. Meiosis increases genetic variation through segregation and independent assortment of genes. So, within a population, there exists some level of genetic variation or differences in traits among individuals. Okay? Genetic variation is due to the presence of alternate forms of a particular gene. Okay, those are called alleles. So you have the mother's genes over here and the father's genes over here. And the alternate forms of a particular gene in this diagram is done by color. Okay, so you could have the red or the purple would be the alternate form for the mother, and then the father's gene is represented by the yellow or the green. And when we get to Punnett squares, you'll see how we represent those as letters, dominant and recessive. So this is an easy diagram done by colors. So in diploid organisms such as humans, each individual carries 2 alleles for one gene. Okay, 2 alleles, red or purple, yellow or green. All right, so the alleles may be the same or they may be different, okay? In this diagram, they're different in each one. Okay, the offspring produced as a result of sexual reproduction resemble but are not resemble but are not identical to their mother or father. So remember how the egg and sperm have half, okay, the chromosome. So the mother still has the option of giving her offspring either the red or the purple, and the sperm has the option, the father has the option of giving their offspring the yellow or the green. So look at this. The child's genes, they get one allele from their mother and one allele from their father. So that's the way we resemble our parents, but we don't look identical to them because we're a mixture of both. So in this example of the child's genes, they obtained a red square allele from their mother and a yellow, all right, circle allele from their father, okay? And they also obtained a purple and a green from one of each, okay? So, and then remember now they're back to the full amount, just how they started off with. Remember the egg and the sperm have half the amount. So this would be representative of 46 total chromosomes, and you get different alleles from the mother and from the father. We're kind of a mixture, combination of both parents' alleles. In addition to segregation and independent assortment, genetic variation can also increase by something called crossing over. It's a process that's actually unique to meiosis. So, crossing over occurs during prophase 1, remember, when two homologous chromosomes pair up. Okay, and they exchange parts of their DNA, all right, as pictured right here. So this green chromosome has a little bit of yellow DNA, and this yellow chromosome has a little bit of green section of its DNA. So crossing over ensures that genetic variation between parents and their offspring is increased. All right, so once again, this image shows that crossing over has occurred between two homologous chromosomes during meiosis. So both chromosomes are now genetically different than they were before. A change in the sequence of an organism's genetic material is known as a mutation. So, mutations can occur randomly through DNA replication when base pairs are either inserted, deleted, or substituted, or they can be caused by environmental factors such as overexposure to radiation or even toxic chemicals. Okay, so if we look at these pictures, here's the original DNA, and there's three different examples here, something called deletion, insertion, and substitution. So each one you can compare to the original DNA strand. All right, so let's look at this deletion one first. It says the second C was deleted. So if we look at our original DNA, here's the second C right here. All right, if we go to the deletion, it has ATC, that's still correct. ATT, so the second C was deleted, so it just moved this T and brought it over. Therefore, it changed the rest of it as well, okay? It removed that C, so I had to bring that T over. So the next group of three is actually TTG, which is down here. And the next group of three isn't even a group of three because one was deleted. So it just ends with two T's. That's called deletion. Now, if we look at the insertion example right here, remember just to compare it to the original DNA strand, it says an extra A was inserted. All right, so it looks like we're good here. That's the same. That's the same. Here's where the extra A was inserted in this area right here. It should be three T's, but instead it inserted an A. All right, so it moved that T to this next spot, changing it to TGT, and then that extra T was left over at the end. That's called insertion. Substitution, here at the end, it says ATC was substituted with GCA. All right, so if we look at our original DNA, it has ATC there, and the next ATC was substituted with GCA. So it just affects this group of three. These are the same. So there's three examples of gene mutations: deletion, insertion, and substitution. A point mutation, it's a type of a gene mutation. A point mutation is a mutation caused by the substitution of a single base pair for another. Okay, so some genetic diseases like cystic fibrosis color blindness, hemophilia, and sickle cell anemia can occur as a result of a point mutation. So this image right here demonstrates an example of a point mutation. Notice how the substitution of a single base pair results in a different amino acid. Remember that codon table. Okay, so this single change results in a different amino acid. So the remainder of the protein sequence remains unchanged. All right. A silent mutation is a specific type of point mutation. So because many amino acids have more than one codon, it is possible for a mutation in a single base pair to have no effect on the polypeptide sequence. So this example shows a silent mutation. Though the second codon, has changed, the second codon has changed. Remember, the group of 3 is a codon, so instead of a U, it's a C here. All right. The amino acid sequence of the resulting protein is the same as the coded for by the normal mRNA strand. So it still codes for this same amino acid. Remember that codon table, and sometimes there are like 3 different codons that coded for the same amino acid. So in this case, that's what it did. And then there's a nonsense mutation where changes in an amino acid codon into a stop codon. Okay, so it changes an amino acid codon into a stop codon. So this causes the normal polypeptide sequence in the resulting protein to be shorter than usual. So almost all of these can lead to a serious genetic condition. A frameshift mutation, it's a gene mutation, it's called a frameshift mutation, is caused by either the insertion, so you're adding, or the deletion, so you're deleting one or more base pairs so that the reading frame of a codon sequence is shifted. Right? So since a codon is a sequence of 3 nucleotides, remember it has the 3 letters together, that code for a specific amino acid, remember your codon table, any insertion or deletion of nucleotide base pairs that are not in multiples of 3 will cause a frame shift mutation. So insertions or deletions in multiples of 3 will cause a protein to be shorter or longer than normal, but the entire sequence of amino acids will not be shifted. So the image shows how insertions and deletions affect a polypeptide sequence. So right here, it's adding, so insertion, a U, all right? So instead of ACU, it's going to add a U in between the A and the C. All right, then it takes that U and it moves it to the next frame. All right, so it shifts everything down. Well, it just happens to be that once it shifted, the UAG coded for a stop codon instead of the correct amino acid, so it made it super short. Okay, and here's an example. Here's your normal mRNA sequence, frameshift, the deletion, right? So it looks like it deleted, it deleted a C. So it's ACU. It deleted this middle C, so it moved to AU, and it brought that A over here to the left. So it shifts everything that way. Okay, and it starts coding for totally different amino acids. Many diseases can be caused by chromosomal mutations. So now we're looking at the chromosome and the mutations within that. So we've got something called non-disjunction, deletion, translocation, inversion, and abnormal karyotypes. So let's take a closer look at each one of those. Non-disjunction occurs when chromosomes do not separate correctly during cell division. The resulting daughter cells will either be missing or having extra copies of a chromosome. So this picture compares a normal cell division All right, to cell division in which non-disjunction has occurred. Some chromosomal abnormalities occur in the sex chromosomes, all right, and we represent the sex chromosomes by X's and Y's, okay? Let's see, so trisomy X, for example, is a type of chromosomal abnormality in which a female has 3 X chromosomes. All right, a female normally would have 2 X chromosomes, but this time in the non-disjunction, the female has 3 X chromosomes. Females with trisomy X often have learning disabilities and may be taller than normal, but they do typically undergo normal sexual development and are able to conceive children. All right. Turner syndrome, on the other hand, is caused by a female only receiving 1 X chromosome. Okay, so remember trisomy, the female had 3 X chromosomes, and Turner syndrome is something where instead of the normal 2 X chromosomes, the female only has 1 X chromosome. Okay, and it can actually result in infertility where they can't have children and other health problems. So both trisomy X, which is this one here, and Turner syndrome are examples of genetic diseases that are caused by non-disjunction. Deletion occurs when part of a chromosome breaks off. Okay, part of the chromosome breaks off and so this is deleted, it breaks off, and it is missing in the offspring. So there's a disease which causes children to have this cat-like cry, and it's caused by the deletion of part of chromosome 5. So that was left off in the offspring. Chromosome translocation occurs when material is exchanged between 2 chromosomes. Okay, here's chromosome A, chromosome B, and they exchange the material, or part of one chromosome becomes fused onto a whole nother chromosome. So some human disorders are caused by chromosome translocation, sometimes infertility and translocation Down syndrome. So translocation Down syndrome occurs because a section of chromosome 21 becomes fused onto another chromosome. Okay, so it accounts for less than 5% of the total cases of Down syndrome reported. Inversion occurs when part of a chromosome breaks off and reattaches in the reverse direction. So here's a chromosome, it breaks in the chromosome and it flips, right? And it's reinserted. So instead of the red and then green, it reinserts with the green on top, okay? All right, abnormal karyotypes. So karyotypes can be used to detect chromosomal mutations and diagnose certain disorders, all right? For example, in humans, Down syndrome is usually caused by an extra copy or trisomy of chromosome 21, resulting in 3 copies of this chromosome being present. So this karyotype is from a human with Down syndrome, and notice that it contains an extra copy of chromosome 21, resulting in a total of 47 chromosomes instead of 46. All right, so that was a lot of info, so make sure to watch this video sometime to learn more about meiosis. Let's do a few practice questions. Number 1, during the process shown below, homologous chromosomes exchange genetic material. What is this process called? So pause it and see if you can remember. So homologous chromosomes contain the same genes, but they do not necessarily contain the same gene sequences. So, for example, a homologous chromosome pair might both contain the gene for eye color, but one chromosome may have the gene sequence for blue eyes, while the other chromosome may have the sequence for brown eyes. So during meiosis, the homologous chromosomes pair and are close together that segments of genetic material actually can be exchanged between these chromosomes. So if you remember correctly, the process is known as crossing over, and it contributes to the genetic variation between organisms. So we're going to go with B. Number 2, a scientist shines low levels of ultraviolet radiation on a dish containing colonies of ruby red bacteria. He then makes several replicate plates from this dish over many generations. He finds that in addition to ruby red color, there are also pink, orange, and yellow bacteria. Which of the following is the best explanation for these findings? So go ahead and pause it and think over the choices. Okay, so mutations are important in how populations change over time because they result in genetic changes to the gene pool. So, in this experiment, ultraviolet radiation induced mutations in the gene for color, which is why new colors of bacteria appeared. Okay, so it looks like we're going to go with D. Mutations occurred in the gene for color of the bacteria, which were passed on to successive generations. Number 3, during sexual reproduction, gametes or sex cells are produced through the process of meiosis. How many chromosomes should be in each cell labeled with an X? Okay, so in this specific species, the cell has 32 chromosomes. And remember, the ones labeled with an X are the gametes, and they should have half. Okay, so half of 32 is what? Okay, so hopefully you went with B, 16. So according to this diagram, the parent cell has 32 chromosomes, right? So each cell labeled with an X should have half that amount, 16. Last one, number 4. Rachel grows pea plants in her backyard. Some of the pea plants have purple flowers, where other pea plants have white flowers. What does this indicate about Rachel's pea plant? So pause it and see what you can think of. All right, genetic variation, like in color, occurs when alleles are randomly assorted during sexual reproduction. So, since each offspring receives a different combination of alleles from the parent organisms, genetic variation results. So, we're going to go with B. The pea plants have genetic variation. They vary in color, whether it's purple or white. Yay! So, after fully covering the topic of genetic variation, you should be able to create a project or presentation or experiment or model or something to let your teacher know that you got this.